I already gave you an introduction about nucleic acids. We talk about the difference between DNA and RNA very briefly, and now I just talk about the difference between replication, transcription, and translation. And now I would like to move to the components of the nucleic acids, and we can discuss in more detail what are the type of sugar that DNA has, what is the structure of the nucleotides, and how you form these phosphodiester bonds. So the polymers of the nucleic acids are polynucleotides, and the monomers will be just the nucleotides. So let me show you what is a nucleotide. Here you can see a polynucleotides, and here you can see at least 3 components. And in this, here we zoom in this small structure, in this small nucleotide that can help us to understand the 3 main components. So a nucleotide will have first a phosphate sugar, second it will have a sugar, in this case a pentose sugar, and finally the third component is a nitrogenous base. And the nitrogenous base is attached to the carbon number 1, and the phosphate and the sugar pentose are attached in the carbon number 5. So to summarize, although, as I said, the polymers of a nucleic acid are called polynucleotides, the monomers are called nucleotides, and each one consists of 3 main components: a nitrogenous base that I showed here, the pentose sugar that I showed here, and finally the phosphate group that is shown here. Another way to divide what is a nucleotide that they give you in your textbook is divided in 2 sections. That is, a nucleotide is a nucleoside plus a phosphate group. So here you find a phosphate group, and the nucleosides will be the fusion between the sugar plus the nitrogenous base. So the portion of the nucleotide without the phosphate group is called the nucleosides. Now let's focus more on the nitrogenous bases. And there are 2 families. One that are the pyrimidines and the other ones that are the purines. And I want that you stop the video and try to compare what is the difference between these 2 families. Do you notice any difference? I know that maybe you already found the difference. Just going to show you again. The pyrimidines and the purines have very different structures. So here you have 1, 2, 3, 4, 5, 6. So here you have a ring of 6 members, like here in all of them you will have 6 members, while here you have 5 and 6, and here you have 2 rings fused of 5 and 6. And this is one key difference. You have a structure of a ring of 6, and this will have 2 rings of 5 and 6. The other difference is that while purines are found in DNA and RNA, pyrimidines have components that are only located in the DNA, are only found in the DNA, and another one, uracil, is only found in RNA. So these are 2 key differences. So let's recap. As I said before, pyrimidines have a single 6-member ring, and the components are cytosine, thymine, and uracil. And the other one is the purines, that I want that you remember, is that it's a 6-member ring fused by a 5-member ring, and this one are, you have adenine and guanine that are located in the DNA and RNA. They are in both, while the uracil is only found in RNA, and the thymine is only found in the DNA. Now let's talk about the difference in the sugars. So in DNA, you have this D because it's deoxyribose. Here you can see the structure. And in RNA, you have the R because of the sugar that is ribose. And now let's try to see the difference. While in the deoxyribose, you have a hydrogen here. In the deoxyribose, you will have an OH. And just remember here, sometimes remember this small difference. Sometimes this question can be in the test, the difference between deoxyribose and the ribose. So we're going to continue talking about the nucleotide polymers, but now we're going to focus on talking about how they link together. I'm sure that you see that there are many covalent bonds between them, but how each nucleotide attached to the next one. So there are two key points here. That here you see a 5' end and a 3' end. So these two, the 5' end and the 3' are fundamental. And in the 5', you're always going to find here 5' is going to be close to the phosphate. And let's go to the zoom of the nucleotide that we see here. Again, this 5' is going to be attached to the nucleotide. On the other hand, and I will change color, the 3' is going to be attached to the OH. Here, when we zoom, you also have the 3' here. And here, as this nucleotide, this 3 carbon is attached to another nucleotide, instead of being an OH, so you have an OH, the OH is replaced. You have an OH here, but instead you have another phosphate. So there are two main components, the 5' and the 3'. The 5' is going to be close to the phosphate and the 3' is going to be close to the OH. So to summarize, here I wanted to show you that let's keep talking about polynucleotides, but now let's talk about how they attach to each other, what type of linkage we find here. And I told you that they have a lot of covalent bonds, but they will have the phosphate on the 5 carbon or on the next. So here in red, you can see all the 5 carbons. I want that you remember. On the other hand, on the 3 carbon of the nucleotide, you will have OH that is on the previous nucleotide. And you can see them here in these black squares. Finally, something that I didn't show here is, but I previously talked, I just remind you that in the carbon number 1', you will always find a nitrogenous base. I'm going to talk about this blue region that is the sugar phosphate backbone that is going to be similar between DNA molecule. So here you will not find any difference. In the similar way, within RNA molecule, this sugar phosphate backbone is also going to be the same. So the difference is not going to come from here. The difference is going to come from the nucleotide, from the sequence of nucleotides. So you will find that each gene will have a different sequence of nucleotides in the DNA and in the RNA. That this will create the uniqueness of each molecule. To summarize, I just told you that first, that these links create a backbone of sugar phosphate units, and here the difference will not come here. The sequence of bases of DNA and RNA will be the one that create the difference for each gene.